This is my video for DNA cyclosequencing. So, I want to bring about 4 or 5 different types of substances. You have your DNA, your primer, your Taq polymerase, your nucleotides, and your dideonucleotides. So, of course, your PCR process is first. So what I did is draw teeth-like structures, and that is your DNA sequence, your DNA sequence, and I denatured them or separate them, and that's your first step or process of PCR. Your next step is to anneal and extend. So you have your primers and your Taqs, and your Taqs will bring about your complementary nucleotides. So let's say my complementary nucleotides were circles. So there's a complementary nucleotide for each. But you do have your dideonucleotides in that test tube or that container. So let's say it stops or connected to this part right here. This is when your Taq knows it stopped bringing in the complementary nucleotides, and that will cause the strand to be cut off. So, like so. But your strands can be in different lengths. So let's say here are my complementary nucleotides, and somehow we have a long strand. Short strands, long strands, it varies. I want to talk about the 4 different types of nucleotides, which are A, C, T, and G. Of course, since there are 4 different types of nucleotides, there are 4 different types of dideonucleotides. In this case, they are abbreviated in DDCTP, DDGTP, DDATP, and so on and so forth. So, your next step is gel electrophoresis. Like I said earlier, there are many different types of lengths of your DNA strands that are multiplied already. So, and there are 4 different types of them. 4 different types of nucleotides. So this is my gel electrophoresis. Since DNA is a negative charge, it would migrate up to down to the positive side. The shorter ones travel the longest, or travel the farthest, and then the longest one travel the shortest. In this case, let's say my A dideonucleotide strand would be green. My C dideonucleotide strand is black. My G dideonucleotide strand is blue, and my T. Let's say my sample made the shortest strand, the T first, and another T, another G, and another A, and then somehow there's a freaking long one and the back. In the end of this structure right here, you have your laser beam. The reason that you need a laser beam is so that once your strands migrate past that that laser beam, it will generate on the computer a set of patterns so you can read your DNA strand. So, like I said, there are 2 red ones, so you would start from the bottom, and another red one, and another blue one, green, and a black one. So, like I said on the top portion right here, I coded T for red. So, you would read this as T, T, G, A, C. And scientists use this to help find different DNA sequences and to compare them. And that is DNA cytosequencing.